Good morning, everybody. Today you are supposed to have self-directed learning or formative assessment. In biochemistry, we are going a bit slow. So instead of those, we will take a lecture class today. Now today we will start a new chapter, amino acid classification and classification. As you know, amino acids are the building blocks of protein. Protein is synthesized from the sequential addition of amino acids. There are about 300 amino acids in the biological system. However, only 20 numbers of amino acids are utilized for protein synthesis. Later on, 2 more amino acids are identified. So altogether, there are 22 numbers of amino acids. Proteins are linear polymers of alpha amino acid, alpha L-alpha amino acids, linked together by covalent peptide bonds. Now, what are amino acids? We know what are alpha amino acid and what are L-alpha amino acids. This is a general structure of this one, amino acid. It has a carbon atom attached with that, an amino group and carboxyl group and R and hydrogen atom. Now, R is a functional group. It can be again hydrogen and other molecule. In nature, amino acids form remains as ionized from the physiological pH. Now, in the structure of amino acid, if we can see that this carbon, it has a 4 different molecule attached to it. Now, when 4 different molecules are attached in the alpha carbon, they can produce optical isomerism. And 2 different optical isomers, they are called enantiomers. And the carbon which can produce this is called chiral carbon. Now, structure of amino acid, it is described in comparison with glyceraldehyde. It is almost similar. Now, when this in glyceraldehyde, when this hydroxyl group is attached to the left, it is called L-glyceride. And when it is attached to the right, it is called D-glyceride. Likewise, in amino acid also, when this amine group, it is attached to the left, it is called L-amino acid. And when it is to the right, it is called D-amino acid. In biological system for protein synthesis, only L-amino acids are used. So, in here we have written L-alpha amino acid. The number of amino acid The carbon atom where carboxyl group and amine group is attached, this is called alpha carbon. Next to alpha carbon is beta, then gamma, delta, epsilon, like that. But in case of numbering of the carbon numerically, when we like to mention, in that case, carbon atom of the carboxyl group is numbered as 1 and the alpha carbon is numbered as 2. Like that, it will go on. So, when we say carbon number 1 means carbon of the carboxyl atom, carboxyl group. When we say the alpha carbon, then this is the carbon where amino group and carboxyl group are attached. So, these are the two different carbon. Now, in case of physiological pH, the amine group, it remains ionized and protonated, and carboxyl group, it remains deprotonated. There are two groups of amino acid, standard amino acids and non-standard amino acids. Standard amino acids are those which are utilized for protein synthesis, have a codon. When you come to the protein synthesis, you will know what is codon. They will have codon, but non-standard amino acids, they will not have codon. But some of the non-standard amino acids also take part in different reactions of biological system. They are D amino acid, non-protein amino acid, and amino acid derivatives. Now, coming to classification of amino acid, they are broadly classified into 4. Based on structural and chemical nature, considering the factors on which the classification will be made, they are based on structural classification, based on polarity, solubility in the water, then based on metabolic fate, whether they can produce glucose or they produce as lipid, and nutritional classification. Here we will find some amino acids which cannot be synthesized in the body, and purely we have to take from the nature, from the outside, to replenish it. So they become essential. They should be taken in food. Now, further classification of amino acid based on the chemical nature and structure, they are aliphatic, having aliphatic side chain with hydroxyl group, side chain with sulfur group, side chain with acidic group, side chain with basic group. Then aromatic amino acid and amino acid. This from classification you will get a short note, definitely you will get. So just try to remember, there is not much to understand. You have to memorize. You have to memorize the thing, and usually it may come as classified amino acid on the basis of structure and chemical nature, or on the basis of nutritional consideration, or metabolic fate. Like that they will give. So you have to memorize the thing. Should not confuse, Now, classification based on structural and chemical nature. First is amino acid with aliphatic side chain. There are 4 amino acids, 5 amino acids: glycine, alanine, valine, leucine, and isoleucine. These An amino acid is denoted by either 3 letters by 3 letter or single letter. Here glycine, for glycine it is GLY and single letter G. Alanine ALA, single letter A. Valine VAL and single letter V. Single letter it should be capital uppercase. Leucine LU capital L, Isoleucine ILE capital I. This is the structure of glycine. Now one thing you note here, this carbon, the 4 functional groups, they are not different. Amine group is there, carboxyl group is there, rest 2 are hydrogen. So this is not a chiral carbon, and glycine will not have optical isomer. That's why optically inactive and it does not have a chiral center. This is the alanine, valine, leucine, isoleucine. Now you can see there is a branch chain in these 3. In valine, leucine and isoleucine. So these 3 are called branch chain amino acid. Now by these mnemonics you can remember Gabli, or you can remember, you can create of your own and remember this. Now these amino acids, lot of numbers are there, different classifications are there, so it will be easier for you if you develop any mnemonics to remember. Now second group, depending on the side chain, is side chain containing hydroxyl group. These are serine, triple letter is SER, capital S for single letter, threonine THR, capital T for single letter, tyrosine TYR, capital letter T. Okay, I will see that again. Now, here is the hydroxyl group in the side chain in place of R. Then here is the side chain. Here is the phenol group and hydroxyl group. Usually, due to this hydroxyl group, they become water soluble. But in the it is written that tyrosine, even though it contains hydroxyl group, it is not readily soluble in water. Next structural classification is side chain with sulfur atom. They are cysteine C, methionine M. Cysteine contains sulfhydryl group here, and methionine contains thioether group. Now another amino acid cysteine, it is the condensation of 2 molecules of cysteine. Here there is a disulfide bond, with that bond they are condensed. Next structural classification, force. This is side chain with acidic group, or they are amides. These are aspartic acid and glutamic acid. And asparagine and glutamic are their amides. Aspartic has 2 carboxyl groups here and here. And glutamine also having 2 carboxyl group. So they are called dicarboxylic acid, monoamino acid. This is only one amino group is there, but 2 carboxyl groups are there. Because of that, they are acidic in nature. Next, aromatic amino acids. Phenylalanine, PHE, but single letter is F. Remember that. 3 letter PHE, single letter capital F. Tyrosine, TYR, single letter T. Tryptophan, TRP, single letter W. So these you have to remember. They are having aromatic groups, benzyl or phenol or indole in their side chain. But proline, it has a amino group. This is the amino group. The proline is having in its side chain. Due to this carboxyl group, presence of carboxyl group, they are water soluble. Now, next consideration is classification based on polarity or solubility water. They are broadly classified into 2 groups, hydrophilic and hydrophobic. Hydrophilic is again negative charge, positively charged or uncharged. Negatively charged are 2 acids, glutamic acid and aspartic acid. Positively charged 3 are there, lysine, arginine and histidine. Uncharged amino acids are those which are soluble in water. They are serine, threonine, tyrosine, asparagine, glutamine, cysteine and glycine. Tyrosine, even though it is not readily soluble in water, but it is to some degree it is soluble in water. That's why it is kept in here. Hydrophobic, non-polar. Aliphatic amino acids, they are alanine, valine, leucine, isoleucine. They are hydrophobic amino acids. And aromatic amino acids, those which are hydrophobic, they are phenylalanine and tryptophan. Proline and methionine, they are also under hydrophobic group. Now classification based on metabolic fate. The carbon chain of the amino acids, they may take part in glucose synthesis or lipid synthesis. Those which take part both in glucose synthesis and lipid synthesis, they are grouped under glucosonic and ketogenic group. These are phenylalanine, isoleucine, tryptophan, tyrosine. You can remember by mnemonics PITT, P-I-T-T, or you can develop of your own. Carbon skeleton of these amino acids, they can go for glucose synthesis also, can synthesize, take part in protein lipid synthesis also. Now, this leucine and lysine, they take part in synthesis of lipid only. So they are ketogenic. Remaining 14 amino acids, they are glucogenic. Next classification is based on nutritional availability. Here are 10 amino acids which the body cannot synthesize. Because of that, they are called essential amino acids. They have to be taken from the dietary source. They are phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, and lysine. For easy remembrance, here one mnemonics is mentioned: private team hall. Private team hall, or you can develop your own in your books also. There are many are there. So according to convenience, you choose one, but do not choose multiple. Then you will confuse in the exam hall, or there will be confusion. So according to availability in the ability of the body to synthesize, these 10 amino acids body cannot synthesize. Now 2 amino acids are there. These two, histidine and arginine, they are called semi-essential amino acids, because in the childhood, infancy and childhood, these amino acids cannot be synthesized by the human. So these amino acids are grouped under essential amino acids. But later on in the adult life, some degree of synthesis can be, body can synthesize these amino acids. But in the exam, if you are asked how many essential amino acids are there, you have to answer 10 itself. 10. There should not be any confusion. It should be 10. And semi-essential out of these 10, histidine and arginine. These two, in the adulthood, to some extent, body can synthesize. That's why they are called semi-essential. Number of essential amino acid, 10. Non-essential amino acid, the body can synthesize. So these are not, even though with food we take, but if required, body can synthesize. The 21st amino acid, Cysteine. In cysteine, there is sulfide group. In cysteine, there is a selenium metal. That is the difference only. And this cysteine is important component of glutathione peroxidase, deiodinase, glycine reductase, thioredoxine reductase. It has a codon also, UGA. The 22nd amino acid is pyrrolysine. Pyrrolysine, the codon is UAZ. It is usually stop codon. During protein synthesis, when DNA polymerase, it comes across this codon, it stops there and protein synthesis is terminated. Okay. Now, this stop codon itself is used as codon of pyrrolysine. There is a separate mechanism by which the polymerase can identify that this amino acid has to be incorporated. This way you will know in your genetics when you read protein biosynthesis. It is similar to lysine, but with an added pyrroline ring linked to the end of the lysine side chain. It is present in methyl transferase enzyme from a methane producing bacteria. Significance of amino acid. Some biologically important compounds which are derived from amino acid. These are from tyrosine, thyroxine, melanin, epinephrine, norepinephrine, dopa. Thyroxine, you know, thyroid hormone. Melanin, responsible for skin pigmentation. Epinephrine, it is responsible for maintaining blood pressure. Norepinephrine also. Dopa, neurotransmitter. Glutamic acid, from glutamic acid is GABA, it is also a neurotransmitter. Tryptophan, from tryptophan it is niacin and serotonin. These are also neurotransmitters. Niacin. It is a vitamin and it acts as cofactor for with enzyme activity. Then glycine, arginine, creatine is produced, responsible for muscle bulk. Glycine and cysteine, bile salt is produced, responsible for lipid soluble vitamin absorption. Glycine, heme. Heme is a metalloprotein. Heme it is present in hemoglobin. Then pyrimidine base, glutamic acid, it is from glutamic acid and aspartic acid. Pyrimidine bases are used in nucleic acid synthesis. It is important component of nucleic acid. Glycine and glutamic acid, aspartic acid, purine bases, pyrimidine bases, purine bases. Then alanine, coenzyme A is produced from alanine. Then histidine, it is histamine is produced. These are the biologically active important compounds, and during the metabolism you will come across again and you will know how they are important. Now, due to the disorder of amino acids, many major diseases of the body are found. This we will discuss during the time of amino acid metabolism. But if I tell now, you won't understand how it is happens. Later on, we will discuss it during the metabolic chapter when we read metabolism of amino acids. So, in summary, we can say amino acids, they are classified into four major groups. Based on classification, based on structure and chemical nature. There are 7 having aliphatic child chain. This is glycine, alanine, valine, leucine, isoleucine. Hydroxyl group containing amino acids, serine and threonine. Sulfur-containing amino acid, cysteine and methionine. Acidic amino acid, aspartic acid, glutamic acid and their amides. Basic amino acid, histidine, arginine, lysine. Aromatic amino acid, phenylalanine, tyrosine, tryptophan and amino acid, proline. Based on polarity, hydrophilic and hydrophobic, based on metabolic rate, ketogenic and glucogenic, and both. Nutritional classification: essential, non-essential, semi-essential. Phenylalanine, valine, tryptophan. Threonine, isoleucine, methionine. Histidine, arginine, leucine, lysine. These are the essential amino acids. Now, let's see the next part, classification of protein. Out of 300 amino acids, only 20 amino acids are taking part in synthesis of protein. Proteins are linear polymers of L-alpha amino acids. They are connected by peptide bonds. Proteins are classified in several ways. Three major types of classifying protein based on their function, chemical nature and solubility of the properties, and nutritional importance. Now, on the basis of function of the protein, it is classified into 8 different classes. Structural protein, taking part in the formation body structure. Storage protein, responsible for storing molecules in the body. Catalytic proteins, these are actually enzymes. Contractile protein, regulatory protein, receptor proteins, protein present in genetic factor, they are called genetic protein. Histones, they are present in DNA. Then defense protein, 9 type of proteins, and transport protein. For remembering, you can just remember like that: SS, CC, RR, then GDT. So, S, structural protein, collagen, example. S, storage protein, ovoalbumin, gluten. Catalytic means enzyme. For example, hexokinase, carbonic anhydrase, alcohol dehydrogenase, etc. Contractile protein, actin, myosin, present in muscle. Regulatory protein, insulin. Now, yes. Insulin, you know, it is a hormone secreted by beta cells of islets of Langerhans, responsible for glucose metabolism. Receptor protein, if this insulin is secreted, then it has to bind to a protein in the cell membrane, which will change the conformation of that protein and allow glucose to enter inside the cell. That is the glucose transporter. Now, if insulin doesn't bind to the receptor, glucose cannot enter. So, for molecule to be transferred from outside cell to inside cell, usually there used to be a receptor. They bind to the receptor and they are taken inside. So these are receptor proteins. There is receptor proteins like G protein. G protein is a kind of receptor protein. Genetic histones, defense protein, immunoglobulins, and transport protein, albumin, transferrin, you know. Next classification on solubility and chemical structure. These are the simple protein, conjugated protein, and derived protein. Simple proteins, they will have only amino acids. Conjugated protein, they will have amino acid attached with a prosthetic group. And derived protein, these are developed, during the digestion of the protein, these either chemically or enzymatically, if it is digested, proteolytic enzyme, then these derived molecules are found. In simple protein, example is again further divided into two, globular protein and scleroprotein. Albumin, for globular protein, it is example is albumin, globulin, globin, glutamine, prolamine, protamines, histones. Usually they are globular in shape and they are water soluble. Scleroproteins, they are fibrous in nature. Usually they are not soluble in water. Collagen, present in tendons. Then elastin, they are present in elastic tissues. Keratin, they are present in hair and skin. Conjugated protein, when amino acid is attached with prosthetic group, they are called conjugated protein. When this prosthetic group is glucose, they are called glycoprotein. Carbohydrate moiety, they are called glycoprotein. Now, if glycoprotein, in glycoprotein, carbohydrate moiety is less than 4%, it comes under glycoprotein, and if it is more than 4%, it comes under mucoprotein. Example of mucoprotein is mucin of saliva. When there is a lipid component with the protein, then it is lipoprotein. Example, HDL, high density lipoprotein, LDL, low density lipoprotein, VLDL, very low density lipoprotein. To have LDL high in the body, it is dangerous because it produces atherosclerosis. Nucleoprotein, it is present in DNA. When nucleotides are there, nucleic acid is there attached with the amino acid. This is called nucleoprotein. When chromogenic particles are there attached with the amino acid, they are called chromoprotein, like cytochrome P54 enzyme. Then when metal is present in the amino acid, it is called metalloprotein. Like carbonic anhydrase enzyme, it contains G as prosthetic group. Now, in derived protein, there are two groups: coagulated protein, proteins, and metaproteins. When partially digested, the second group is produced. They are peptones, polypeptides, peptides, and proteases. Now, in classification, basing on the nutritional consideration, proteins can be classified into complete protein, incomplete protein, and poor proteins. Complete proteins are those which contains all the amino acids required for the body. Example is albumin, milk casein. Incomplete protein, there will be absence of one essential protein in those proteins. They are incomplete proteins. Like in cereal proteins, there is absence of lysine. In pulses, there is absence of methionine. So, significance is, when taken, those foods having incomplete protein, we have to mix. We have to mix those two so that the lacking amino acid is filled up from the other. Cereal and pulses, they have to mix together, so the one is replenished by the other. Now, poor proteins are those, there is absence of more than one number of amino acid which is required for the body. Example is zein in corn protein, zein. It is devoid of tryptophan and lysine. Now, here structure of protein. Protein structure are of 4 levels. Primary structure, secondary structure, tertiary structure and quaternary structure. In primary structure, only amino acid chain is there. Only amino acid chain. The important thing to remember in the primary structure is that the amino acids has to be in a particular sequence linked by peptide bonds. And the number of amino acid for a given protein is fixed. So, number of amino acid, sequence of amino acid, these two things are very important for the primary protein. They are connected, amino acids are connected by covalent bond, covalent peptide bond, and occasionally there is disulfide bond. Now in insulin, there are two side chains, two chains of insulin polypeptide chains, A and B. It starts with glycine, A chain, this is B chain. It starts with phenylalanine. Now, there is a disulfide bond with the 7 amino acid of A chain and 7 amino acid of B chain. Again, there is a disulfide bond between the 20th number of amino acid of A chain and 19th number of amino acid, the B chain. Again, there is a disulfide bond between amino acid 6 and 11. 6, amino acid 6 and 11. So, this disulfide bond and polypeptide bonds, they stabilize the protein in primary group. Now, in case of secondary structure, four types of secondary structures are there. Most common are alpha helical structure, beta plated sheet, and with these turns are there, bands are there, loops are there. Now, alpha helical structure is the most common form of secondary structure. It is the polypeptide chain. It is folded spirally and it is stabilized by covalent bond, hydrogen bond, and destabilized by proline and glycine. The helix is right-handed helix. It is like that. This is the axis, and helix, it will turn like that. Right-handed helix. Now, in case of one turn, say this is turned like that and it comes here. So it forms a 360-degree turn. Now, total number of amino acid involved in this 360-degree turn is 3.6, not even 4 amino acids. 3.6 amino acids are there in one turn, from here to here. Distance between amino acids are 0.15 nanometer. Hydrogen bond between the carboxyl group of the first amino acid with the nitrogen of fourth amino acid. Stability of the helix is given by hydrogen bond, electrostatic bond, van der Waals forces. Next type of structure is beta helix. In beta helix helical structure, there are two polypeptide chains arranged in the same direction. Their amino terminals will be in same side, and carboxyl terminal will be in the other side, in the same side, both of them in the same side. And stability is given by hydrogen bond, electrostatic bond, then van der Waals forces between the two polypeptide chains. Usually they are arranged in zigzag manner. They are arranged in zigzag manner. They will they will get arranged on not only two polypeptide chains, there may be so many chains. They are stabilized by hydrogen bond, then electrostatic bond, van der Waals forces. They may be arranged in the opposite direction. That means so two polypeptide chains, they are arranged in opposite direction. So interchain hydrogen bond, electrostatic bond, van der Waals forces, with that they are stabilized. Now turn, loops, bands, they are usually these type of structures, they remain in collecting these two polypeptide chain. There will be a band here, or turn, or loop. Tertiary structure, this is the three-dimensional structure. So this is the primary structure. Secondary structure is coming like that. For example, I am just drawing so it may be connected by band or loop. This is the secondary structure, beta plated sheet, and tertiary structure, this is another alpha helical loop. Now this primary structure is again folded on it to produce secondary structure. Two-dimensional structure it will produce. So this is the tertiary structure. Here also the configuration is stabilized by hydrostatic bond, van der Waals forces. Almost in all configurations, same type of electrostatic force, hydrogen bond, then van der Waals forces. These are the stabilizing bonds. Quaternary structure. Now when this is called unit or subunit. So to become functional, some protein will have 2 subunits, some will have 4 subunits. I mentioned during transport aquaporin, it has 4 subunits. So one subunit, another subunit, like the 4 subunits, they are kept together to form the quaternary structure. If we separate these subunits, then protein becomes functionally inactive. So rest of the